So I call upon the chairpersons for the next session, Dr. Sushil Kotru, Dr. Pawan Sharma, Dr. Ratnakar Sahu, and Dr. Mohammad Hafizur. Request the chairpersons to introduce the first speaker and start with session 2.
Good afternoon to all. We'll start with the first presentation of today's evening session. It will be on biosimilar insulin. The topic is the first interchangeable biosimilar insulin, improving affordability among diabetes patients. It will be presented by Dr. Richa Chaturvedi. She's here. Over to you, Dr. Richa.
Thank you, chairpersons, for the kind introduction. So, welcome to this session on biosimilarity and interchangeability, particularly in context with insulin glargine. A study published in Lancet in 2018 found that the amount of insulin required to effectively treat adults with type 2 diabetes patients is likely to increase by 20% over the next 12 years. And without major improvements in insulin access, insulin would be beyond the reach of approximately half of the adults with type 2 diabetes mellitus who require it. And these findings are of particular concerns for regions like Africa and Asia, which the study predicts would have the largest unmet need for insulin if the insulin access remains at current level. And despite the United Nations commitment to effectively treat NCDs and to make anti-diabetic drugs universally available, insulin is scarce, and at times patients unnecessarily struggle to really have access to it. It is in bridging this gap between the projected demand and supply that biosimilar insulins can play an important role. As of now, in US market, there are more than 30 biosimilar products. And of all these biosimilar products, Biocon's biosimilar insulin glargine is the first biosimilar product that has obtained the designation of interchangeability by FDA. So what is biosimilarity? What is a biosimilar product? A biosimilar product is a biological product which is highly similar but not exact. A biosimilar product is a biological product which is highly similar but not exactly identical to the reference licensed product. And what is a biological product? A biological product is a complex protein which is manufactured with the help of a living organism, a yeast, bacteria, or an animal cell. And what is interchangeability? An interchangeable biosimilar product is a product which can be substituted for the reference licensed product at the pharmacy level without the intervention of the prescriber, because it has been demonstrated in adequately powered studies to be almost similar to the reference licensed products. Now, the word interchangeability sounds like a very simple term, simple word, but there are laws to define it. And FDA in US requires separate switch studies to demonstrate interchangeability before it can be given interchangeability tag, whereas the biosimilar product can be substituted for the reference licensed product. So biosimilar products, they have had a bumpy ride. Back in 90s, late 90s, small difference in packaging of biosimilar erythropoietins, it caused exaggerated incidence of PRCA, pure red cell aplasia. And the incidence peaked in 2001, whereas there were more than 170 cases of pure red cell aplasia with a minor packaging difference in the manufacturing of biosimilar erythropoietins. Now this made the world to look with caution at biosimilar products, and it also prompted the regulatory agencies to formulate stringent guidelines to approve biosimilar products. And bowing to these guidelines, different processes, improvements in clinical monitoring programs were designed, and by 2004, the incidence of pure red cell aplasia came down drastically. It went down by 80% by 2004. Another example is of Marvel Insulin Limited, which was a UK-based company, and it submitted its application for marketing authorization of Marvel insulins to CHMP, Committee for Medicinal Products for Human Use. But the application was turned down because there were not adequate comparability exercises and inadequate data, substandard data. So finally, Marvel Insulins withdrew its application for marketing authorization of insulins. There are differences between a bike, a car, and aeroplane. And there are differences between generics and biologics. Generics are small molecules like statins, like metformin, aspirin. So these are formed in a predictable manner, in a controlled manner, in laboratory using a chemical recipe. These are small molecules, whereas biologics, these are large, complex protein molecules which have, because they are proteins and because they are formed with the help of living organisms, it changes the whole equation. Whenever a living organism is involved, you can't be 100% sure, you can't 100% predict what the final product would be. So they have to be adequately tested for immunogenicity, immunologic reactions. So separate studies are required for biologics. They are large, complex protein molecules. What is a biosimilar insulin? A biosimilar insulin is a biological drug that is similar to an existing insulin, but cannot be considered an exact copy of the original branded licensed insulin because of the different manufacturing process. Now, this manufacturing process, this is a proprietary knowledge known only to the manufacturer. This is not made public. So two insulins made by two different manufacturers cannot be 100% identical. And as stated, they involve a living organism. They cannot be 100% identical. There will be some differences, but some differences in inert, inactive components are allowed, but there should not be clinically meaningful differences. And these are produced from living cells or organisms using biotechnology. They show more heterogeneity. So the gene which is inserted inside the vector, it will control the process till the stage of translation. But there would be post-translational changes. Because of that, glycosylation would occur. The protein molecule, insulin molecule will fold on itself, will change the shape, and this may bring about certain changes in the overall clinical profile of the insulin. And again, this needs to be tested. So what they are not, biosimilar insulins, they are not biomimics or biocopies. It's not that they can bypass the regulatory pathway. Regulatory pathway is abbreviated, yes, but they cannot altogether escape it. There are different ways for their regulatory approval, as we would discuss. And they are not biobetters. They are not claiming to incorporate any better gene or anything to make them better. They are almost similar to the reference licensed products. And they are, as stated, they are not generic drugs. Generic drugs are small molecule drugs which can be made in a laboratory using a chemical recipe in a predictable and controlled manner. So one unifying theme in biosimilar industry is the process is the product. If the process is right, final product would be right. And it's a step-by-step process, and every step of the process has to undergo the same high standard of making. So how the insulin is produced? So insulin gene is isolated from a gene donor. It is combined with plasmid, and then this is inserted inside a vector, which could be a yeast, a bacteria, or an animal cell line. And this vector, yeast or bacteria or animal cell line, this acts as mini factory, and it spits out copies of insulin, which is then purified, harvested, and packaged. And as you can see later on, the protein folds on itself also, changes the shape, which is a post-translational step of the process. And the insulin manufacturing facilities, they need to undergo periodic quality checks by regulatory authorities like FDA or European Medical Agency. In biosimilar development, in vitro techniques to demonstrate the similarity to the reference product is something of paramount importance. There are various complex techniques to ensure equivalent surface charge, purity, activity, concentration, and sizes are involved. And a biosimilar product has to undergo all these complex stages. Now, looking at the regulatory pathway for a reference product and for biosimilar product. The original drug development is a new drug development plan where there is a the regulatory requirements are different. There would be some in vitro studies, something like proof of concept study, and then there will be in vivo study in the living cells. Then there will be usual PK/PD study, what the drug does to the body and what the body does to the drug. And then once sort of we have some proof of safety and efficacy, it has to be it has to be tested in humans, and large clinical trials are required for the development of the originator molecule. Whereas a regulatory pathway for a biosimilar molecule, it is abbreviated. There will be large in vitro studies, because that is the main thing that they have to prove, that their molecule is almost similar to the reference licensed product. And once that has been proven, we know what the reference product does, and the reference product has already been tested in humans and shown the effect. So the most important exercise in a biosimilar drug development is in vitro study step, which we, as we saw in the last slide, it is done very meticulously. Then there would be some in vivo studies, and there will be PK/PD studies. And FDA does not require a huge complement of clinical trials, because the reference product has already been tested. So FDA does not require that the drug be a lot of human testing, a lot of clinical trials are done. And this particular thing, this helps in cutting down the cost. It expedites the regulatory approval, and it eases the market entry for the manufacturer. So physician's perspective, what do we expect from a biosimilar insulin? Yes, it should be safe, it should be effective, it should not have any untoward adverse reactions there, and because this is a protein, there should not be untested immune reactions. And of course, there should be some cost advantage to our patients. So that is about Biocon. Global trials have been done, and Biocon's insulin glargine has approvals by major regulatory agencies like FDA, European Medical Agency, and India approval. Available in different regions by different names, Basalog in India and Semglee in US. Looking at non-inferiority study for granting biosimilarity tag to Biocon's insulin glargine. So basically there are 3 studies. InStride 1 is done in type 1 diabetes, InStride 2 in type 2 diabetes. Then FDA requires separate switch studies to grant interchangeability designation. For that they have InStride 3 studies. So InStride 1 is multi-center, open-label, non-inferiority trial to compare the efficacy and safety of Biocon's insulin glargine with reference insulin glargine. Approximately 548 patients here, 52-week study, and primary endpoint was HbA1c changes from baseline to 24 weeks. The study met its primary endpoint. There were no statistically significant differences in HbA1c levels achieved, and the secondary endpoints were changes in fasting plasma glucose, total insulin dosage used, SMBG levels, hypoglycemia, nocturnal as well as anytime hypoglycemia, and immune reactions, and they were found to be comparable in both the arms of this study. InStride 2 study was done in type 2 diabetes patients. It was a non-inferiority study to compare the efficacy and safety of Biocon's insulin glargine with reference glargine in type 2 diabetes mellitus, both in insulin naive patients as well as insulin non-naive patients. It was very important to study this new molecule in insulin naive patients, where prior insulin exposure will not confound the immune reactions caused by this new protein. So this was done in around 560 patients, 24-week study, and primary endpoint was change in HbA1c at 24 weeks, and the endpoint was met. There were no statistically significant difference in HbA1c levels achieved. And secondary endpoints were changes in FPG levels, changes in total insulin dosages, and anytime in nocturnal hypoglycemia as well as in immune reactions, and they were found to be comparable in both the arms. FDA required separate switch studies to grant interchangeability designation to a biosimilar product. European medical agencies, they have different rules. They leave it to their member countries to grant the tag of interchangeability. But FDA requires switch studies. So InStride 3 was a switch study. The patient population was taken from InStride 1 study, type 1 diabetes patients who were on reference insulin glargine arm, and it was a 36-week study in arm B, lower side, as you can see. These people were around 63 patients. These people were on reference insulin, licensed insulin glargine throughout the period of 36 weeks. Whereas in arm A, switch sequence was used. For initial 12 weeks, patients were shifted to Biocon's insulin glargine. Next 12 weeks, patient came back on reference licensed insulin glargine, and the last 12 weeks they came back on Biocon's insulin glargine. So this was switch sequence for 36 weeks. And the primary endpoint was change in HbA1c from baseline to week 36, and change in HbA1c from baseline to week 36 with Biocon's insulin glargine, that is in switch sequence, was equivalent to reference insulin glargine. And the safety endpoints were anytime hypoglycemia and nocturnal hypoglycemia, and these were comparable in both the arms. And immune data by collecting antibody levels was also analyzed, and they were comparable in both the arms. So it demonstrated equivalent efficacy, safety, and immunogenicity, demonstrating that patients taking reference insulin glargine can safely switch to Biocon's insulin glargine. So after this switch study, this interchangeability study, FDA granted the interchangeability designation to Biocon's insulin glargine, and approval of this interchangeable insulin glargine can provide patients with additional safe, high quality, and potentially cost-effective option for treating diabetes. While granting interchangeability designation to Biocon's insulin glargine, FDA noted today's approval of first interchangeable biosimilar product furthers FDA's long-standing commitment to support a competitive marketplace for biological products, and ultimately empowers patients to increase the access to safe, effective, and high quality insulins. And because the studies have demonstrated the interchangeability benefit, it can be provided, it can be available at a lower cost and will be as safe and effective as the licensed product is. So approved and quality biosimilars can be beneficial for the patients in terms of safety, efficacy, and hopefully in terms of affordability and accessibility. When a diabetes prescription becomes longer, the cost of individual medicines, individual products becomes a very important factor, and that is how we have to justify writing so many medications still, expecting patients to afford it, because there is no insurance system in our country. Biosimilar insulin glargine is up to 48% economical than reference insulin glargine. Vials are 48% more affordable than reference product, and cartridges are up to 23-24% more affordable than the reference product. So when a patent expires after years of exclusivity, it is time to include similars. In 2009, February, Fisher from European Biopharmaceutical Enterprises, while presenting the dossier looking at the scientific aspects for formulating the guidelines for biosimilar approval, once again emphasized that the process is important, and minor differences in process may lead to remarkable differences in clinical profile, and things that look alike at low resolution may become less similar or increase analytical sensitivity. Therefore, standardized processes and continued vigilance are important to ensure safety and efficacy of biosimilar products. So to summarize, systematically developed and approved biosimilar insulins can bridge the gap between demand and supply of insulin. Biocon has a formidable track record in insulin manufacturing and in making insulin accessible to our patient. It has a global presence and aims to be insulin provider to 1 in 5 insulin dependent patients. Thank you very much for joining me in this session.
Thank you, Dr. Richa, for sticking to the time, and you have made this presentation very simple and lucid. You have shown that these biosimilar insulins, they have been US FDA approved, and they are safe, and they have the same efficacy as the innovator brand, and they are affordable also, which is very important from Indian perspective.
From patient perspective, yes, especially in a country like us where patient has to pay funds for.
Because of paucity of time, we can take 1 or 2 questions, not more than that. 1 question.
Yeah, sure. Can I put the question?
Yeah.
Thank you, ma'am. Thank you very much for your wonderful presentation. You clearly alluded that what is interchangeable, that is where the patient gets into the pharmacy, where the pharmacists take the decision to refill with the biosimilar drug. That's clear. Now, the point here is that who are the right patients that you really see who could be put this kind of the interchangeable stuff?
As I pointed out, when the diabetes prescription becomes longer, for example, suppose a patient is taking oral hypoglycemic agents and we are giving glargine insulin. And now that we find that the HbA1c is not well within the range, it is not meeting the target, and we have to add another insulin. We are adding another analog, something like short-acting analog. We are making either a basal plus regimen or a basal bolus regimen, and we are adding a analog, something like Lispro. Now patient has to pay for both. So what we can adjust in the same cost, patient can buy Lispro and a less costly glargine. So I think in such scenario where the total, we are increasing the medication, yet the total cost of prescription for the patient remains same, I think that would be a useful scenario for our patients.
Thank you, ma'am. Thank you very much.